Nadia, and I welcome you to my short presentation about heterogeneous biological networks with the stringApp in Cytoscape. If that's the first time you hear about the stringApp, I'll start with a short introduction. High-throughput technologies nowadays produce long lists of genes or proteins that are associated with a certain phenotype or disease of interest. Such technologies can be, for example, proteomics or transcriptomics, sequencing, and so on. Usually, one thing that one can do with this list of proteins is go to a public database like String and find out if these proteins have something in common, if they're connected via protein-protein interactions. However, web interfaces often have limitations due to the size of the input list and also the flexibility you have for analyzing and visualizing the data, which is why you can use also the standalone tool Cytoscape to visualize and analyze the networks of your lists, like I did in this example. Here, additionally, I also highlighted the up and down regulated proteins using the values that come from the original table. The stringApp can be found on the Cytoscape App Store, like all the other Cytoscape apps. Everything is for free. And besides fetching string networks into Cytoscape, it can also do a lot of other fun stuff, like, for example, retrieve functional enrichment, create a network for disease of interest, annotate your proteins with tissue expression or subcellular localization. These are some of the things that have been around for quite some time now, and in the last few months we have worked on some new features that I would like to share with you. For example, you can not only start with a list of proteins, but also with a list of your own interactions that come from an affinity purification mass spectrometry experiment, like in this case. Here, I show you the supplementary table from the study of Gordon et al. In Nature from 2020, where they basically took most of the SARS-CoV-2 proteins, they cloned them, tagged them, and expressed them in human cells in order to find which human proteins they are likely to physically interact with. So we have two important columns here, the baits, these are the SARS-CoV proteins, and the preys are the human proteins that interact with them. We can take this table, load it into Cytoscape, and then we get this network as a result. In this network, each star basically represents one SARS-CoV protein and all the human proteins it interacts with according to the experiment. Then we can change this network by stringifying it and basically converting all the human proteins into string nodes and getting, for example, tissue expression about them. We can use this to check whether the proteins are expressed in the lungs, in blood. And we can also go one step further and retrieve all the physical interactions between the human proteins that interact with the SARS-CoV-2 proteins. And what you can see here is basically that proteins that interact with different SARS-CoV-2 proteins also interact with each other and most likely build a physical complex together. Last but not least, one can use different ways in Cytoscape to visualize this network and expand and highlight some things. For example, in this case, I highlighted the SARS-CoV proteins, making these red diamonds and changing the labels a little bit. Virus-host interactions are just one example for interspecies interactions, but we also interested in host-parasite interactions, especially in eukaryotes, but also other organisms, because they cause several different diseases, and looking into the interactions will help us understand these diseases better. This is why we are working on putting both known and predicted functional and physical interactions between eukaryotic parasites and their hosts into the stringApp. We do that by collecting and aggregating evidence from text mining, experiments, expert knowledge and autology transfer. And we also, as usual for string interactions, calibrate the scores based on the KEGG pathways in complex plot-out databases. As a result, we can create a host-parasite interaction network that has edges that are scored with confidence scores about how likely the proteins are to interact with each other. Here we see an example for a functional interaction network between human proteins, the gray nodes, and proteins from different parasites, or the colorful nodes. And what you can see here is that basically in several cases, we can see that proteins from different parasites interact with the same set of human proteins. To summarize some of the new and exciting stringApp features, we can actually choose between two different types of networks. We can not only have a functional interaction network, but we can also have a physical interaction network. We can access all the 14,000 organisms in the current string version. And we also provide interspecies interaction between, for example, host and parasite pairs or virus host pairs. You can also come with your own interaction network and combine this with string using stringify and compare and integrate it with what is available in the string database and associated databases. And you can also do functional enrichment for whole 16 different categories, the usual thing like gene ontology and KEGG pathways, but also for tissues, diseases, and phenotypes. Thank you, and looking forward to hearing from you if you're interested to know more about the stringApp.